how do you ask a mouse if it's hallucinating? You really can't. A rodent cannot pull up a chair, sit across from a clinician, and just articulate some complex, systematized delusion about the CIA tracking its movements.
No, obviously not.
Right. But in trying to solve that completely impossible problem, we've actually accidentally mapped the biological engines of human fear, addiction, and despair. At its core, psychiatry is the study of how brain, mind, and experience interact, and crucially, how that incredibly delicate process can go wrong.
Right.
But when we try to approximate the human mind in an animal, it's essentially like trying to understand the vast, unpredictable open ocean by studying a controlled, shallow tide pool.
I love that. And that is exactly the tension we navigate every single day in the lab. I mean, the tide pool operates on the exact same fundamental physical principles, right? You've got water, gravity, salinity, the basic biological drives for survival. So a tide pool isolates those specific mechanisms beautifully. You can watch a crab scavenge or a barnacle filter feed without a hurricane getting in the way.
Yeah, but the tide pool completely lacks the crushing pressures, the terrifying lightless depths, and the sweeping currents of the open sea. Animal models are our psychiatric tide pools. They isolate the mechanism, but they can't possibly capture the existential entirety of the human experience.
And to be fair, we really shouldn't expect them to. Abstract thought, complex language, the uniquely human capacity for self-reflection, and the punishing rumination that often stems from it, those are features of the open ocean. But underneath all that, our subcortical structures are ancient.
The evolutionary engine room.
Exactly. The amygdala, the hippocampus, the striatum, those are highly conserved across species. The hardware running a mouse's fear response is stunningly similar to the hardware running yours.
So, welcome to this deep dive. We are going straight to the core of how we translate a mouse's freezing behavior, or a dog's sudden sleep attack, into actionable neurobiological insights for human mental illness. And let's unpack the core problem straight away, because a lot of people look at this field and think it's inherently a fool's errand. Looking for schizophrenia in a rat feels a bit like looking for a Wi-Fi signal in a toaster.
Historically, yes, that was a massive stumbling block. For a long time, the field was completely obsessed with creating a schizophrenic rat or a depressed mouse.
Right.
Researchers were trying to model entire psychiatric disorders, but there was a paradigm shift a couple of decades ago when we collectively admitted defeat on that front. We realized you simply cannot model entire human psychiatric disorders in animals.
Because the human diseases are incredibly messy.
Highly heterogeneous, yes. The diagnostic criteria we use for humans, the DSM or ICD, they're based on subjective, self-reported symptoms. Two humans can both be diagnosed with major depressive disorder and share almost zero overlapping symptoms.
Oh, absolutely. One can't sleep, won't eat, and is agitated. The other sleeps 16 hours a day, overeats, and can't get out of bed.
Precisely. To create a genetic mouse line that encapsulates all of that contradiction is scientifically impossible.
So instead of looking for schizophrenia as a whole, we break it down into Lego bricks. We look for a specific symptom, like an inability to filter out background noise.
We call those symptom domains. Things like negative valence, which covers fear and anxiety, or positive valence, covering reward and addiction, or cognitive deficits. These domains cross diagnostic boundaries. So a deficit in working memory is a hallmark of schizophrenia, but it's also present in severe depression and ADHD.
Makes sense.
By looking for well-defined phenotypes, observable traits with known biological bases and strong genetic homologies between humans and animals, we can do highly precise science.
Which brings us to the ultimate question of whether that science is actually any good. How do we know if the model works? It all comes down to validity. There's this sort of holy trinity of validity in the literature. First, we have face validity, which seems like the most intuitive one.
It is. Face validity just asks on its face, does the animal's behavior physically resemble the human symptom? If we're modeling panic and a rat exhibits frantic, darting flight behavior when a predator gets close, well, that looks like human panic. It has strong face validity.
But appearances are notoriously deceptive in neurobiology. I mean, I might be crying because I'm depressed, or I might be crying because I'm chopping an onion. The output is exactly the same, but the mechanism is vastly different, which is why we need the second pillar, which is construct validity.
Yes, construct validity is much more rigorous. It demands that the animal model reproduces the actual etiology, the underlying biological root cause of the human disorder.
Can you give an example of that?
Sure. Let's say we find a specific, highly penetrant genetic mutation in human families that causes a neurodevelopmental disorder. If we use gene editing, to put that exact human mutation into a mouse's DNA, and the mouse develops the corresponding cognitive deficits.
Then you've nailed it.
Exactly. That model has exceptional construct validity. We aren't just mimicking the symptom, we've recreated the biological spark.
And then there's predictive validity, which is essentially what the pharmaceutical industry lives and dies by.
Right. Predictive validity evaluates whether a manipulation in the animal, usually giving it a drug, accurately predicts clinical effectiveness in humans. If a novel compound reduces freezing behavior in a mouse, does it actually cure anxiety in a human patient?
Yeah.
A model with high predictive validity has very low false positive and false negative rates.
And here's where it gets really interesting and, frankly, quite dangerous for clinical reasoning. Because you can have a test with fantastic face validity, but absolutely terrible predictive validity. Think of it like trying to measure wind speed with a thermometer. The thermometer is a great tool. It measures weather. But if you're trying to figure out if a hurricane is coming, the temperature reading is going to give you a massive false negative.
And that happens all the time in preclinical psychopharmacology. An animal might look sad, it stops moving, it stops eating, so a researcher gives it a new drug, the animal starts moving again, and they rush to publish that they've cured depression.
But then it fails in humans.
Exactly. If the drug consistently fails in human clinical trials, the model lacked predictive validity. We have to be incredibly disciplined about not taking animal behaviors purely at face value.
Let's apply that discipline right now, actually. I'm going to jump into our first major domain, which is negative valence. This is the dark side of the emotional spectrum. Fear, anxiety, hopelessness. It's rooted heavily in those ancient subcortical survival circuits you mentioned earlier.
Specifically, we're talking about structures like the amygdala, the periaqueductal gray, and the bed nucleus of the striatum terminalis. These areas govern emotional states of aversiveness or discomfort.
We colloquially use fear and anxiety interchangeably, but neurologically they're entirely different beasts. It's all about the proximity of the threat. Let's use a metaphor. Fear is when you're sitting in your office and suddenly thick black smoke starts pouring under the door. You are in immediate acute danger. Anxiety is when you're sitting in your office, you hear a fire alarm 3 blocks away, and you spend the next hour wondering if the wind is going to shift in your direction.
That distinction maps perfectly onto the ethological findings in rodents. The brain processes distal threats, the fire alarm 3 blocks away, completely differently than acute, imminent threats. When a threat is distal, the animal engages in anxiety behaviors like freezing, concealment, and risk assessment.
Keeping a low profile.
Yes. The animal lowers its posture to avoid detection, but when the threat is acute, the smoke under the door, the animal transitions immediately into explosive flight or defensive panic attacks.
So how do we trigger that acute panic in a lab setting?
The most direct ethological method is the defense test battery. You place a rat in a long circular runway. Then an experimenter introduces a predator, sometimes a live cat, sometimes a robotic predator, that begins closing in on the rat.
A robotic predator?
Yeah.
That's brilliant, but I assume that's primarily for construct validity. So the threat is exactly the same every single time.
It ensures absolute standardization of the stimulus, yes. As the robotic predator approaches, we quantify the rat's flight response. If we suddenly block the runway, creating an unavoidable threat, the rat's brain shifts gears again. It goes from flight to defensive threat and attack.
It turns around and fights.
Exactly. This model has excellent face validity, and more importantly, drugs like benzodiazepines, which we use to treat human panic attacks, reliably reduce this runway flight behavior.
But dealing with live predators, or complex circular runways, introduces a lot of mechanical noise. There's a much more elegant, purely neurological way to induce panic, is there?
Yes, bypassing the external environment entirely and going straight to the brainstem. We can electrically stimulate the dorsal periaqueductal gray or the DPAG.
What exactly is happening in the DPAG?
The DPAG harbors a highly sensitive hypoxia alarm system. It's basically the brain's internal sensor for suffocation. When you stimulate it, the rat exhibits profound, explosive, panic-like responses. It's not just running, it's a frantic, desperate escape behavior.
And this is where the construct validity shines, because human panic disorder isn't just one monolithic thing. Some people get heart palpitations, some get dizzy, but there is a specific respiratory subtype of panic disorder where patients feel this overwhelming, terrifying sense of smothering or air hunger.
Exactly. The DPAG stimulation in rats perfectly models that specific respiratory subtype in humans. It shows us that this type of panic isn't a cognitive failing, it's a false alarm in the brain's deepest suffocation circuitry.
Okay, let's shift from that unconditioned raw panic to conditioned fear. because this is the gateway into learning theory, and it's how we start to unpick the neurobiology of trauma and PTSD.
Conditioned fear relies on standard Pavlovian principles. You take a neutral conditioned stimulus, let's say a specific audio tone. You play the tone and immediately follow it with an aversive, unconditioned stimulus, which is usually a very mild electrical foot shock.
It only takes a few pairings for the amygdala to wire those two things together. Tone equals shock.
It's extremely rapid, robust learning. Once the association is formed, we can measure the learned fear. A common way is fear-potentiated startle. If you play a loud noise to a rat, it'll flinch. That's the baseline acoustic startle reflex. But if you play that same loud noise while the fear-conditioned tone is playing in the background, the rat will flinch significantly more violently. The underlying state of fear potentiates the acoustic startle.
We see that in humans with PTSD. A car backfiring might make me jump, but it makes a combat veteran hit the deck. The baseline threat arousal just magnifies the startle.
Absolutely. But the truly vital clinical application here isn't just understanding how fear is learned, it's understanding how it's extinguished.
Extinction. This is the biological bedrock of exposure therapy.
If you take that fear-conditioned rat and you play the tone over and over again, 50 times maybe, but you never deliver the foot shock, the brain starts to learn a new rule. The rat learns that the tone is now safe. The conditioned freezing behavior gradually diminishes. This is extinction.
And crucially, extinction is not forgetting. The rat hasn't forgotten the shock. It's actively laying down a new inhibitory memory trace right over the old fear memory.
That's a critical distinction, and because it's an active learning process, it relies heavily on neuroplasticity, specifically the NMDA glutamate receptors in the amygdala. This gave us a massive pharmacological target. Researchers reason that if we can boost NMDA receptor function, maybe we can speed up this extinction learning.
Which led to the trials with D-cycloserine.
Yes, D-cycloserine is a partial agonist at the NMDA receptor. In the animal models, giving D-cycloserine before extinction training significantly accelerated the reduction in fear. It basically made the exposure therapy work faster and stick better.
And that translated directly to the clinic for human-specific phobias and social anxiety. Giving a patient D-cycloserine just before an exposure therapy session, like putting them in a glass elevator if they have a fear of heights, actually improves the clinical outcome. Though I know the literature is a bit messier when it comes to complex PTSD.
Well, yes, the clinical reality of PTSD often involves multiple compounded traumas over years, which is vastly more complex than a single tone shock pairing in a pristine lab environment.
Let's look at more generalized anxiety, where there isn't a specific tone or a specific trauma. The classic models here revolve around approach avoidance conflicts.
Rodents are natural foragers, so they possess an innate drive to explore novel environments for food, but they are also prey animals. Out in the wild, an open, brightly lit, elevated space is exactly where a hawk will snatch them. So their neurobiology is constantly weighing the drive to explore against the drive to hide, and we exploit that conflict.
The elevated plus maze is the poster child for this. It's exactly what it sounds like. A raised platform shaped like a plus sign. Two arms have high, dark walls, so they feel safe and enclosed. The other two arms are completely open, essentially just diving boards into the abyss. You put a mouse in the center. A calm mouse will eventually creep out onto the open arms to explore. An anxious mouse will stay huddled in the dark, walled arms.
We use similar paradigms like the light-dark box, where the animal chooses between a brightly illuminated chamber and a pitch-black one, or the open field test, where we measure how much time the animal spends clinging to the perimeter walls versus venturing into the exposed center.
Okay, let's unpack this for a second because I want to push back on these approach avoidance models. The literature explicitly notes that these specific mazes were primarily developed to validate the effects of benzodiazepines, like Valium or Xanax. If our primary tool for measuring anxiety was built entirely around detecting one specific class of drugs, aren't we just trapped in a massive confirmation bias loop?
It's one of the most glaring translational failures in our field. You've hit the nail on the head. These tests are exquisitely sensitive to the GABAergic mechanisms of benzodiazepines. If you give a rat a benzo, it will happily stroll out onto the open arms of the plus maze. But consider SSRI selective serotonin reuptake inhibitors,
Like fluoxetine or sertraline, which are the absolute gold standard first-line pharmacological treatments for human generalized anxiety disorder.
Right. But if you give an SSRI to a rat and put it in the elevated plus maze, it often fails. The SSRI does not consistently increase time spent in the open arms. If an alien scientist landed on Earth and only used the elevated plus maze to screen drugs, they would literally throw away every SSRI, completely missing the most effective anti-anxiety medications we possess.
Because the thermometer doesn't measure wind speed. We're using the wrong biological ruler.
Yeah.
So how do we bypass this? How do we test anxiety without relying on a test biased toward benzos?
We look to different behavioral outputs. The marble burying test is one. You place a mouse in a cage filled with deep bedding and 24 glass marbles. Mice will naturally engage in defensive burying of novel, unfamiliar objects.
So an anxious mouse will frantically bury every single marble, whereas a calm mouse will just ignore them.
That's the theory. But the construct validity there is hotly debated. Are we measuring anxiety toward the object? Or are we measuring a repetitive, compulsive phenotype? Some argue that drugs reducing marble burying are actually just increasing passive freezing behavior, which is really just a different flavor of anxiety.
A better alternative seems to be the shock probe burying test.
Much better. You introduce a small electrified probe into the cage. The rat approaches, sniffs it, receives a mild shock, and retreats. Then, the rat will actively use its forepaws to kick bedding over the probe to bury it. This is a highly specific, active defensive response to a confirmed, localized threat. It's much cleaner than marble burying.
But there's another confound we haven't mentioned, which is sedation. Benzodiazepines make you sleepy. If a rat is just sitting in the corner of an open field, is it less anxious, or is it just too sedated to move?
Locomotor confounds ruin a lot of data.
Yeah.
To bypass behavior entirely, we use stress-induced hyperthermia.
Taking their temperature.
Exactly. When a mammal experiences acute stress, the sympathetic nervous system triggers a slight but measurable increase in core body temperature. Often, just the minor stress of the researcher picking the rat up to insert a rectal thermometer is enough to trigger the response. We can measure this physiological spike independently of whether the rat is running around or sedated in a corner. It's a pure autonomic readout.
I want to scale up from rodents for a moment. Social anxiety is profoundly complex. It's the fear of judgment, of observation. How do you model that?
We have to move up the evolutionary ladder to non-human primates, like macaques or marmosets, because their social hierarchies are much closer to ours. The classic model is the human intruder test.
It sounds like a home invasion.
It's remarkably simple, actually. You have a primate in its enclosure. A human researcher, who the primate has never seen before, steps into the room and stands perfectly still, maintaining a specific profile, purposefully avoiding direct eye contact.
Just looming in their peripheral vision.
And for a primate, that ambiguous presence triggers profound behavioral inhibition. They freeze, but more tellingly, they begin exhibiting displacement behaviors. They'll start scratching themselves obsessively or engaging in frantic auto-grooming. It's the primate equivalent of a socially anxious human nervously biting their nails or checking their phone at a party. It responds beautifully to clinical anxiolytics.
But the human intruder introduces noise. I mean, I might be taller than you or wear a different color lab coat. To remove that experimenter variable, there's a newer touchscreen paradigm.
Yes. This leverages their ethology much better. The monkey is trained to touch specific shapes on a computer screen to run a juice reward. It's a calm, routine task. But randomly, the screen will flash a high-definition image of an unknown, dominant male monkey of the same species staring directly at them.
Which is a severe social threat challenge in primate language.
The reaction is instantaneous. You measure their behavioral inhibition. Do they physically recoil from the screen? How long does it take them to overcome the fear and touch the screen again to get the juice? It directly taps into the neurobiology of social dominance and submission without the noise of a human experimenter in the room.
It's incredible how precise we can get. Let's pivot slightly within negative valence, moving from the hyperarousal of anxiety to the hypoarousal of depression. We're talking about hopelessness, despair, reduced effort. How do you quantify despair in a mouse?
The most ubiquitous acute screens are the forced swim test for rats and the tail suspension test for mice. The forced swim test involves placing a rodent in a shear-walled cylinder of water. They can't touch the bottom, and they can't climb out.
So it's an inescapable stressor.
Right. Initially, the animal swims frantically, desperately trying to find an escape route. But after a few minutes, it stops struggling. It adopts a posture of immobility, making only the absolute minimal movements required to keep its nose above the waterline.
And historically, we've interpreted that transition from active struggling to passive floating as behavioral despair. learned helplessness. The animal essentially decides, nothing I do matters, so I give up.
And the predictive validity is why this test is so famous. If you administer a clinically effective antidepressant like an SSRI or a tricyclic, the animal will spend significantly more time actively swimming and trying to climb before it finally gives up.
But let's look at the construct validity because there is a huge debate here. Is the rat really despairing? Or is it just exhibiting high-level cognitive adaptation? I mean, if I'm thrown into the middle of the ocean, thrashing around is going to drain my energy and drown me faster. Floating conserves energy. Maybe the rat isn't depressed. Maybe it's just smart.
That critique is heavily cited. Are we measuring despair or are we measuring adaptive energy conservation? But regardless of the philosophical interpretation of the behavior, the pharmacological reality remains. The test reliably detects drugs that augment monoamine transmission. What's fascinating is how we use it today. Standard SSRIs require weeks of chronic dosing to work in humans, but they work acutely in the forced swim test. Now, researchers are using this exact same test to map the mechanisms of rapid-acting agents like ketamine and psilocybin.
Proving that a single dose of ketamine, acting on glutamate rather than serotonin, can induce an immediate, resilient, active coping strategy in the animal that lasts for days.
Exactly. But despair is only one half of the depressive phenotype. We also have to measure anhedonia, the profound loss of the capacity to feel joy or seek reward.
We measure that by looking at what the animal usually loves. The sucrose preference test is the gold standard. Rodents have a massive sweet tooth. If you put 2 bottles in their cage, one with plain water, one with sugar water, they will almost exclusively drink the sugar water. But when they are in a depressive-like state, that preference completely collapses. They'll drink from both bottles equally or barely drink at all. They just don't care anymore.
The progressive ratio test takes that a step further by measuring not just preference, but motivation. How hard will you work for the reward? The rat presses a lever to get a sugar pellet. The first pellet costs 1 press, the next costs 2. Then 5, 10, 20, 50.
So we're looking for the break point.
The exact moment the neurobiological cost-benefit analysis shifts, and the animal decides the physical effort is no longer worth the dopamine hit. In anhedonic animals, that break point plummets. They give up much earlier.
But how do we get the animal to that anhedonic state in the first place? Acute stressors like the forced swim test don't really capture the chronic, grinding daily friction that usually precipitates clinical depression in humans.
For that, we rely on chronic stress models. The most robust is the chronic social defeat model. This leverages the intense territoriality of male mice. You take a subject mouse and you place him into the home cage of a much larger, highly aggressive, dominant male, usually a specifically bred aggressive strain.
So the subject mouse is immediately attacked and physically subordinated.
Yes. The attack is stopped before serious injury occurs, but then the subject mouse is forced to live in that same cage for 24 hours, separated only by a perforated plexiglass divider. He can't be bitten, but he can smell, see, and hear the aggressor constantly. It is an environment of inescapable chronic psychosocial stress. You repeat this with a new aggressor every day for 10 days.
And the behavioral changes in the subject mouse are just catastrophic.
They exhibit profound social avoidance. If you put a completely novel, non-aggressive mouse in their arena, the defeated mouse will just cower in the far corner. Their sucrose preference drops, their progressive ratio breakpoints plummet, they are neurobiologically depressed.
Here's where it gets really interesting, and honestly represents one of the most frustrating historical blind spots in neuroscience. For decades, this incredibly powerful, robust model for depression was fundamentally mathematically flawed because it only worked on males.
The territorial aggression required to make the model work is a male-specific ethological trait. Female mice, generally speaking, do not display or elicit that type of fierce, cage-defending aggression when confronted with a novel female conspecific. So for decades, researchers simply excluded females from chronic social defeat studies.
Which is mind-boggling when you look at the epidemiology. Women experience clinical depression at roughly twice the rate of men. We were studying the neurobiology of a disease predominantly affecting women by exclusively looking at male brains.
And we know now that the molecular reality of depression is sexually dimorphic. The transcriptomic profiles, the actual genes being turned on and off in the brain during chronic stress, are vastly different between male and female mice. By excluding females, we were literally missing half the biological picture.
Thankfully, the literature outlines some brilliant ethological workarounds that have recently solved this.
It requires tricking the social circuitry, One method involves applying male odorant urine or specific pheromones to the female subject before placing her in the aggressor's cage. The male's olfactory system registers a male rival, and he attacks.
But there's a more organic adaptation involving maternal aggression, right?
Yes, you house a female aggressor with a castrated male. Because the male is castrated, they don't breed, but they form a tight pair bond. The female establishes that cage as her territory and views the castrated male as her mate. If you then drop a female intruder into that cage, the resident female's maternal territory defense circuits fire rapidly. She will fiercely attack the female intruder.
Finally, allowing us to subject female mice to chronic social defeat and opening up an entirely new frontier studying sex-specific neurobiology and depression. It's a perfect example of how the design of the model dictates the absolute limits of our knowledge. Let's cross the spectrum now, moving from negative valence to positive valence.
Yeah. Positive valence covers emotional states that are intrinsically attractive or rewarding. In a healthy brain, this system drives us to eat, mate, and survive. But in psychiatric disorders, we are usually looking at what happens when this dopamine-driven system becomes completely unmoored, leading to destructive hyperarousal or addiction.
The ultimate expression of hyperarousal is mania in bipolar disorder. And again, this seems impossible to model. I can't imagine a rat experiencing grandiosity or maxing out a credit card or talking a mile a minute.
We can't model the cognitive grandiosity, but we can model the biological triggers and the behavioral output. If you look at human bipolar patients, severe sleep disturbance is one of the most reliable triggers for a manic episode. So researchers leverage that using a sleep deprivation paradigm.
This is the flower pot technique.
It's conceptually simple. You place a rodent on a very small platform like an inverted flower pot surrounded by a pool of water. The platform is just big enough for the rat to sit, eat, and groom, but when a mammal enters REM sleep, they experience atonia, complete muscle relaxation.
So the moment the rat hits REM sleep, it goes limp, its nose hits the water, and it wakes up.
It completely deprives them of REM sleep. If you enforce this for 72 hours, the animal's neurobiology snaps. It enters a state that remarkably mimics an acute manic episode. The rat becomes hyperactive, exhibiting stereotypic repetitive pacing. Even when you take them off the platform and allow them to sleep, they exhibit severe insomnia. They become highly aggressive, and they exhibit profound hypersexuality.
All classic hallmarks of human mania. And the predictive validity is the clincher here.
It is. If you treat these sleep-deprived rats chronically with mood stabilizers, specifically lithium, these manic behaviors are almost entirely blocked. The model proves that the biological link between sleep architecture and mood regulation is conserved across species.
Let's drill down into impulsivity. The literature divides this into impulsive action and impulsive choice. And clinically, it's vital to separate the two. Impulsive action is essentially a failure of the brakes. You know you shouldn't do something, but the prepotent motor response fires before the prefrontal cortex can veto it.
We measure impulsive action using the stop signal reaction time task, the SSRT. It's a beautifully precise test of inhibitory control. The rat is trained to press a lever quickly when a go signal flashes to get a reward. But on a small fraction of the trials, a no-go signal flashes milliseconds after the go signal. The rat has to abruptly cancel its initiated motor response.
It's like swinging a baseball bat and trying to stop mid-swing when you realize it's a curveball.
Exactly. We calculate how long of a delay between the two signals the rat can tolerate and still successfully hit the brakes. We also use the 5-choice serial reaction time task, which tests sustained spatial attention. The rat is facing an array of 5 holes. It has to wait for a light to flash in one of the holes, then poke its nose in to get a treat. If it gets impatient and pokes its nose in before light flashes, that's a premature response, a failure of impulsive action.
And drugs like atomoxetine or methylphenidate, which we use for ADHD in humans, significantly reduce those premature responses in rats.
Then we have impulsive choice. This is cognitive impulsivity. It's the classic delayed discounting paradigm. Would you rather have a small reward right now or a massive reward later? An impulsive brain heavily discounts the value of future rewards. It can't wait.
But the textbook raises a fascinating translational roadblock here. In human studies, I can offer you 10 pounds today or 100 pounds next year. I can just verbally explain the future. You can't explain the concept of next week to a macaque monkey.
That's a huge point. In animal models, the subject must experientially learn the delay. They have to press the large reward lever hundreds of times and sit there experiencing the 30-second delay over and over until the brain maps the temporal cost. So when we compare rodent data to human data, We have to be careful. We can only accurately compare it to human tasks where the human subjects also had to sit in a room and actually experience the delay in real time rather than just answering a hypothetical questionnaire about the future.
Let's link impulsivity to compulsion. Because on the ward, they can look identical. A patient keeps gambling or a patient keeps washing their hands. But the mechanism is totally different. Impulsivity is acting without thinking. Compulsion is feeling physically forced to act despite knowing the action is senseless. How do we isolate compulsion in a lab?
We look for perseverative responding, the inability to break a cognitive loop. The reversal learning paradigm isolates this perfectly. You train an animal on a simple rule. The left lever gives food. The right lever does nothing. They learn it perfectly. Then, without any warning, you reverse the contingencies. Now, the right lever gives food.
An impulsive animal might just start mashing both levers randomly in frustration.
But the compulsive animal will exhibit perseveration. It will continue to press the left lever 50, 100 times, completely ignoring the overwhelming empirical evidence that it's no longer working. The behavioral flexibility circuit in the prefrontal cortex is completely jammed.
We also see this rigid compulsion in eating disorders. The textbook details activity-based anorexia, which has to be one of the most deeply counterintuitive models in all of science.
It defies basic survival logic. If you restrict a mammal's food intake, its evolutionary programming should force it to conserve energy, to sleep, to lower its metabolic rate. In the activity-based anorexia model, you restrict a rat's access to food to only a short window each day, usually just an hour or two. But you also give them free access to a running wheel.
And paradoxically, instead of resting, they start running.
They run excessively. And the more they run, the less they eat during their designated window. It creates a lethal neurobiological feedback loop. The starvation triggers hyperactivity, which suppresses appetite, causing more starvation. If researchers don't intervene, the rats will literally run themselves to death.
It's a harrowing mirror of the lethal drive we see in severe human anorexia nervosa, where hyperactivity and severe caloric restriction lock together. But because we don't have any genuinely effective, targeted pharmacotherapies for anorexia yet, we can't fully validate the predictive validity of the model.
On the other side of the eating disorder spectrum, we model binge eating using the cafeteria diet. If you give rodents access to highly palatable, high-fat, high-carbohydrate human foods, essentially junk food, and restrict their access to it, they will gorge. The vital finding here is that chronic exposure to this cafeteria diet causes a severe downregulation of the dopamine receptors in the striatum,
Which is the exact same neurological signature we see in drug addiction. The brain gets flooded with so much dopamine from the junk food that it removes receptors to protect itself, meaning the animal has to eat even more next time just to feel normal. Which brings us right to the door of substance use disorders, the hijacked brain.
The modern clinical framework views substance use disorder entirely as an acquired disease of neuroplasticity. The addictive substance fundamentally rewires the reward circuitry, causing the brain to radically overvalue the drug at the total expense of natural rewards like food, water, or social connection.
The easiest way to measure drug reward is conditioned place preference, or CPP. You put a mouse in a box with two distinct chambers. One has striped walls, one has spotted walls. Day 1, you lock them in the striped room and inject them with cocaine. Day 2, you lock them in the spotted room and inject them with saline. After a few days of this, you open the partition and let them choose where to hang out.
And overwhelmingly, they will choose the striped room. The brain has associated those specific spatial cues with the massive dopamine spike of the drug.
But the text is quite blunt about CPP having limited construct validity for human addiction. Why?
Because it's a totally passive model. The experimenter is injecting the drug. The animal is just a recipient. But human addiction is defined by active, desperate, operant drug-seeking behavior. The patient is doing the work to acquire the drug.
So we move to active self-administration.
Yes. We surgically implant intravenous catheters into rodents or primates. We place them in operant chambers and train them that pressing a lever delivers a microinfusion of a drug directly into their bloodstream.
And the translation here is stark. Animals will readily, eagerly self-administer almost every single drug that humans abuse. Cocaine, heroin, methamphetamine, nicotine, alcohol.
Interestingly, they will not self-administer classic hallucinogens like LSD or psilocybin, which maps perfectly onto the fact that those drugs do not drive compulsive addiction in humans.
But pressing a lever for a hit of cocaine doesn't equal addiction. Lots of humans try cocaine and don't become addicted. We have to model the clinical reality of the disease, which is the loss of control. The progressive ratio test comes back into play here. We measure the breakpoint, Will the rat press the lever 100 times for a single infusion? 500 times? 1,000 times?
It measures the escalating motivational value of the drug. But the most clinically relevant model, the one with the highest construct validity for the sheer destruction of addiction, is modeling compulsive use despite severe negative consequences.
This is the foot shock paradigm.
Yes. Once an animal has established a strong self-administration habit, you change the rules. Now, pressing the lever delivers the hit of cocaine, but it simultaneously delivers a painful electrical shock to their feet.
A healthy animal, or one that has only had brief exposure to the drug, will stop immediately. The neurobiological logic says the pain isn't worth the reward.
Exactly. But an addicted animal, one whose prefrontal cortex has been downregulated by chronic extended drug access, cannot stop. The compulsive drive overrides the pain signals. They will continue to press the lever, enduring severe, repeated shocks just to maintain their drug levels. It perfectly models the human patient who continues to inject heroin despite losing their marriage, their career, their health, and their freedom.
And the tragedy of addiction doesn't stop when the drug stops. We have to model relapse, extinction, and reinstatement.
You force the animal into abstinence by removing the drug. The lever stops working. Eventually, the lever pressing extinguishes. Then we study what triggers reinstatement, what causes them to relapse and start frantically pressing the lever again.
And the triggers are identical to human triggers. A stressor, like a random foot shock, will trigger relapse. A small priming dose of the drug given by the researcher. Or, most powerfully, just the drug-paired cue. Just flashing the light that used to turn on when the drug was delivered is enough to send the animal into a frenzy of drug seeking.
And this leads to perhaps the most terrifying finding in addiction neuroscience, the incubation of craving.
You would logically think that the longer someone is abstinent, the weaker their craving gets. Time heals all wounds.
But the neuroplasticity of addiction works in reverse. If you measure how hard an animal will work for the drug after 1 day of abstinence, it's high. But if you force them into abstinence for 30 days or 60 days and then expose them to a trigger, the magnitude of their relapse, the sheer violence of their lever pressing, is exponentially higher. The craving actually intubates and amplifies over time.
Which explains why a patient could be clean for a year, walk past the pub they used to drink at, and suddenly experience a craving so intense it feels like a physical blow. The brain was quietly sharpening that knife in the background. Let's shift gears now into the third broad domain: cognition, social functioning, and sleep.
Cognitive deficits are often the most debilitating treatment-resistant symptoms in psychiatry, especially in schizophrenia. To understand cognition, we have to start at the very bottom, before a thought even forms. Sensory gating, pre-attentive processing.
Your brain is constantly bombarded with sensory data, the hum of the fridge, the feeling of your socks. If your brain processed all of it at full volume, you'd go mad, so it filters it out.
We measure this filtering using prepulse inhibition, or PPI. It relies on the acoustic startle reflex. If I sneak up behind you and detonate a firecracker, you will exhibit a massive full-body startle response. But if I tap you firmly on the shoulder a fraction of a second before I detonate it, your brain registers the tap, the prepulse. It uses that minor stimulus to warn the system and actively dampen the startle response to the subsequent loud noise. You'll still jump, but it will be heavily inhibited.
In a healthy brain, that prepulse inhibition is intact. But in patients with schizophrenia, particularly those actively experiencing psychosis, this early sensory filtering is fundamentally broken.
The prepulse does nothing. The loud noise is processed at maximum intensity. The brain is utterly overwhelmed by incoming stimuli, which many theorists believe is the foundational mechanism that generates delusions. The brain is desperately trying to make sense of a tsunami of unfiltered sensory data.
And PPI is completely conserved across species. We can put a rat in a soundproof box, play a quiet tone, then a loud tone, and measure the flinch. If we give them an antipsychotic, like haloperidol or clozapine, we can watch it restore their prepulse inhibition in real time.
Moving up from sensory gating, we look at working memory, the ability to hold and manipulate information transiently. This is heavily reliant on the prefrontal cortex. In rodents, we use the radial arm maze. Imagine a central platform with 8 distinct arms radiating outward. Food is placed at the end of each arm. An efficient rat will visit each arm exactly once.
It has to hold a real-time map in its head, like, I've cleared arms 1, 3, and 4, don't go back there. If it re-enters an arm it's already emptied, that is quantified as a working memory error.
But translating working memory from rodents to humans is fraught. Rodents are brilliant for early screening, but evolutionarily, our prefrontal cortices diverged tens of millions of years ago. The rodent prefrontal architecture lacks the granular complexity of the primate brain.
Which is why primate models are irreplaceable here. Monkeys can perform delayed match-to-sample tasks. A screen drops, they see an object, the screen goes opaque for 20 seconds, and then they have to pick the matching object from a complex lineup.
And because primate prefrontal homology is so tight to humans, the data translates flawlessly. In fact, the CAN-TAB cognitive assessment system, which clinicians use in hospitals every day to assess human dementia and schizophrenia, was directly reverse-engineered from these specific primate touchscreen tasks.
Let's talk about declarative memory, spatial memory. The textbook brings up the absolute titan of behavioral neuroscience, the Morris Water Maze.
It is the most famous spatial memory test in the world. You have a large circular pool filled with opaque water. Just beneath the surface, completely invisible to the swimming animal, is a small resting platform. You drop a rodent in. It swims frantically until it accidentally bumps into the hidden platform and climbs up. Over multiple days, the rodent uses visual cues on the walls of the laboratory to triangulate the exact spatial location of that hidden platform.
You test their memory by taking the platform out and watching to see if they swim in tight focus circles exactly where the platform used to be. It's incredibly elegant. But I want to push back on this. The water maze is universally used, but it has a colossal biological confound. You are dropping a rat into a pool of water where it cannot touch the bottom. It thinks it is going to drown. It is swimming for its life. Doesn't that acute survival panic completely spike their cortisol levels? Are we actually measuring spatial memory, or are we measuring how severe stress degrades hippocampal function?
That is a phenomenal critique, and it is the exact reason many researchers are moving away from the water maze. The physical exhaustion of swimming, the thermal stress of the water temperature, the massive sympathetic nervous system activation, these are all massive confounding variables. The stress of impending drowning can alter the results entirely independently of the actual memory circuit you are trying to study.
So what's the alternative? How do you test spatial memory without the drowning confound?
We use the Barnes maze. It's a large, elevated circular table with 20 circular holes drilled around the perimeter. Underneath 19 of the holes, there's just a drop to the floor. But underneath one specific hole is a dark, enclosed, safe escape box.
You put the mouse in the brightly lit center of the table.
Right. And think about the ethology of a mouse. It is a prey animal. Its entire evolutionary biology screams that standing in a bright open space means a hawk is going to eat it. It desperately wants to find a dark, enclosed space, so it explores the edges until it finds the escape box.
But crucially, it isn't drowning. It's uncomfortable. It's motivated by a natural ethological aversion to open spaces, but it isn't an acute survival panic.
Precisely. Over several trials, using the exact same spatial cues around the room, the mouse learns to run in a straight line directly to the correct hole. It measures the exact same hippocampal-dependent spatial declarative memory as the water maze, but it completely removes the confounding variable of acute survival stress. It's a much cleaner, far superior model.
Let's touch briefly on social functioning. Attachment. We mentioned primate social anxiety, but what about the positive side? Affiliation. Love.
For that, we owe an enormous debt to the prairie vole. The vast majority of rodent species are highly promiscuous, they mate and move on. But the prairie vole is uniquely monogamous. They form profound, lifelong pair bonds. They share parenting duties. If a male prairie vole's partner dies, he will often refuse to take a new mate, exhibiting behaviors that strikingly resemble human grief.
It's heartbreaking, but scientifically, it's a goldmine.
It allowed us to decode the neurobiology of attachment. By studying prairie voles, we discovered the absolute criticality of oxytocin and vasopressin in cementing social bonds. If you block oxytocin receptors in a prairie vole's brain, they become just as promiscuous as a standard mouse. It proved that attachment isn't just a psychological construct, it's a hardwired neurochemical circuit.
Finally, in this domain, we have sleep and arousal. And this is where we have to tell the story of narcolepsy, because it is arguably the single greatest triumph of construct validity in the history of our field.
Human narcolepsy is devastating. It's characterized by excessive daytime sleepiness and sudden, uncontrollable sleep attacks. Most notably, patients experience cataplexy, a sudden, complete loss of voluntary muscle tone while they are fully awake, usually triggered by a strong, positive emotion like laughing.
For a long time, we had no idea what caused it. But in the late 20th century, researchers identified a specific colony of dogs primarily Doberman pinschers and Labradors, that exhibited naturally occurring narcolepsy.
It was startling to watch. The dogs would be playing or about to eat a treat, experiencing high positive emotion, and they would suddenly collapse to the floor in a cataplectic fit, fully conscious but paralyzed. Researchers developed the food-elicited cataplexy test to quantify it.
But the real breakthrough was the genetics.
Yes. Because these dogs naturally possessed the disease, researchers systematically sequenced their genome to find the flaw. And in doing so, they discovered an entirely unknown neurobiological system, the orexin receptor system, also known as hypocretin. They proved that a specific genetic mutation causing a loss of function in the orexin system was the root cause of the canine narcolepsy.
And when they took that canine data back to humans, they struck gold. We now know definitively that human narcolepsy type 1 is caused by the autoimmune destruction of orexin-producing neurons in the hypothalamus.
It is the ultimate translational triumph, discovering the exact biological mechanism in an animal that maps flawlessly onto the human etiology. It led to genetic mouse models, and it is directly responsible for the entirely new classes of targeted orexin antagonist sleep medications we use in the clinic today.
Which brings us to a critical juncture: the integration, the clinical reasoning. Because the narcolepsy story is beautiful, but it's the exception. It is not the rule. This leads us to the preclinical bottleneck. If our models are as sophisticated as we've just described, if we can map fear extinction and working memory and impulsivity with such surgical precision, why do the vast majority of psychiatric drugs that cure mice fail completely when they reach human clinical trials? This is the crux of the entire field. The bottleneck exists because of how we define human disease. We diagnose humans using syndromal behavioral clusters, the DSM and the ICD. We say a patient has major depressive disorder because they check 5 out of 9 symptom boxes on a clinician's clipboard. But the biological reality beneath those checkboxes is vastly, wildly heterogeneous.
Exactly. Let's say we have 2 patients in the waiting room, both diagnosed with major depression. Patient A's depression is driven by severe neuroinflammation cytokine cascades. Patient B's depression is driven by chronic stress, altering their epigenetic transcription in the hippocampus. Clinically, they look identical. Now, a pharmaceutical company develops a brilliant new drug validated in the chronic social defeat mouse model. The drug perfectly reverses the epigenetic stress adaptations.
So they take that drug, they bypass the mice, and they run a massive phase 3 clinical trial on 1,000 depressed human patients.
And the trial fails. The drug is shelved. Why? Because the drug only works on the underlying biology of patient B. It does absolutely nothing for patient A, whose depression is inflammatory. But because our clinical trials group them all together under the monolithic syndromal umbrella of major depression, the overall statistical efficacy of the drug looks no better than a placebo.
The syndromal diagnosis is masking the biological precision of the drug. The drug didn't fail, our diagnostic manual failed, so how do we fix it?
The future relies on precision medicine and diversity. On the precision front, tools like CRISPR-Cas9 gene editing allow us to create highly specific genetic models that map onto actual human biology, not just human behavioral checklists. RNA sequencing and proteomic screens are refining our construct validity to an unprecedented degree.
And on the diversity front, the textbook is adamant. We cannot keep using exclusively male, genetically identical, highly inbred strains of mice.
It guarantees clinical failure. Human populations are wildly heterogeneous. If we ignore sex as a fundamental biological variable, as we did with the social defeat model for decades, we guarantee that our findings will only ever apply to half the human population. True clinical reasoning requires us to understand individual, population-based, and species-specific differences.
Let's pressure test all of this. I want to throw some rapid-fire clinical scenarios at you based on everything we've just covered to see how these models actually apply to reasoning on the ward or in the lab.
Let's do it. Focus on the mechanisms.
Scenario 1. A researcher develops a new rat model testing a novel addictive compound. When the drug is abruptly removed, the rat exhibits severe, quantifiable physical withdrawal symptoms, shaking, temperature dysregulation, mirroring human physical dependence perfectly. What specific type of validity is the researcher predominantly claiming here? Pause the audio here if you want to think it through.
That is a classic claim of face validity. On sheer observation, the behavior, the physical manifestation of the withdrawal looks identical to the human symptom it is intended to model. It doesn't guarantee the neurochemistry matches, but it looks right.
Scenario 2. You're consulting on a neurobiology paper. The team is electrically stimulating the dorsal periaqueductal gray, the DPA, in rat. Which specific human psychiatric phenomenon are they most accurately modeling? Pause the audio here if you want to think it through.
They are modeling the respiratory subtype of panic disorder. Because the deep hay harbors that specific hypoxia-sensitive alarm system, it perfectly mirrors panic attacks characterized by a sense of smothering, asphyxiation, or profound air hunger.
Scenario 3. A massive pharmaceutical company synthesizes a novel compound. It demonstrates incredibly potent anxiolytic effects in the elevated plus maze. The rats are exploring the open arms fearlessly. But it fails completely in Phase 2 clinical trials for human generalized anxiety disorder. What known limitation of the plus maze likely explains this multi-million-pound failure? Pause the audio here if you want to think it through.
The failure stems from the test's inherent historical bias. The elevated plus maze was primarily developed and validated to detect the GABAergic mechanisms of benzodiazepines. It is notoriously insensitive to other mechanisms. For instance, it frequently fails to detect the effects of SSRIs. The novel compound likely worked via a non-benzodiazepine mechanism that the maze was biologically blind to.
Scenario 4. A lab is studying declarative spatial memory, but they decide to explicitly abandon the Morris water maze in favor of the Barnes maze. Why would they make that specific methodological choice? Pause the audio here if you want to think it through.
To eliminate a massive confounding variable, which is acute survival stress. The water maze induces the panic of swimming and potential drowning, which spikes cortisol and alters hippocampal function. The Barnes maze achieves the exact same spatial memory assessment, but relies merely on the rodent's natural ethological aversion to open, brightly lit spaces, providing a much cleaner, unstressed assessment of memory.
Scenario 5. If you want to model the true clinical reality of human substance use disorder, the absolute loss of control, which behavioral paradigm gives you the highest construct validity? Pause the audio here if you want to think it through.
Drug self-administration maintained despite aversive foot shock. That active model captures the core, destructive compulsion of addiction, the willingness of the subject to continue seeking the drug even when it guarantees immediate, severe, and escalating physical punishment.
Scenario 6, last one. The study of naturally occurring narcolepsy in canine models, the Dobermans collapsing when excited, led to the breakthrough discovery of which crucial neurobiological system? Pause the audio here if you want to think it through.
It led directly to the discovery of the orexin or hypocretin receptor system. It remains the gold standard example of an animal model providing perfect construct validity that translated flawlessly into human pathophysiology and subsequent drug development.
Brilliant. So what does this all mean? I want to leave you with a final provocative thought to mull over as you head back to the clinic or step back into the lab. We spend a massive amount of time wringing our hands over the preclinical bottleneck. We lament the fact that our animal models, with their pristine CRISPR knockouts in perfectly controlled environments, are consistently highlighting discrete, reliable biological mechanisms. But then the drugs fail in human trials.
We constantly blame the animals. We say the mice are failing to model human disease.
Exactly.
Yeah.
But maybe the animals aren't failing at all. Maybe our human diagnostic manuals are failing to map onto biological reality. Perhaps the mice are trying to tell us that major depressive disorder isn't one disease. Maybe it's a dozen different, distinct biological lesions all masquerading under the exact same syndromal trenchcoat. If we actually listen to the precision of the animal models, it might just force us to completely burn down and rewrite how we diagnose human beings.
And that translation from the tide pool to the open ocean is the ultimate goal of psychiatric neuroscience.
Here are the details of this episode.
Animal models in psychiatry. Byline. Approximating the human mind, what models reveal and what they cannot.